In this third video, I'm picking up on the third and fourth roles of alveolar epithelial type II cells, releasing superoxide dismutase 3 and catalase to the outside of alveolar epithelial cells. We will look at how superoxide is made from oxygen using the perspective of redox chemistry and enzyme mechanisms and quantum mechanics of molecular orbitals, because these perspectives are needed to understand superoxide dismutase 3 and catalase. Superoxide is made by the redox reaction in which O2 gains an electron or is reduced. In redox reactions, there is an exchange of one or more electrons. Recall that reduction is gain of an electron. The O2 molecule can gain a total of 4 electrons. Stepwise, this first makes superoxide, then hydrogen peroxide, then the hydroxyl free radical, and finally water. Linus Pauling and Newman named the free radical negative anion made from reacting molten potassium with oxygen superoxide. Hund's rule says for degenerate orbitals, ones with identical energy levels, the electrons are added one to each orbital with the same spin before adding a second electron to any of the degenerate orbitals. Here we arrive at the result that O2 is a dual free radical. This is a really big deal because it explains how oxygen can be thermodynamically activated, have a lot of energy stored in the form of oxygen, that is, and kinetically stable. If it were not kinetically stable, we would burst into flames. It turns out that in quantum mechanics that there is a law of conservation of angular momentum that says in a closed or isolated system, the total angular momentum is conserved, so spin is conserved like charge is conserved. O2 is forbidden to react with any of the normal biomolecules such as fats, lipids, amino acids, carbohydrates, etc. These molecules have only paired electrons. At the end of such a reaction, the sum of the angular momentum and the spin orbital momentum would not be conserved. Think of hemoglobin and rust. Iron, like other transition elements, is filling up the d orbital and has unpaired electrons in the d orbital. In other words, iron is a free radical. It turns out that O2 can react with free radicals without breaking the law of conservation of angular momentum. So oxygen is carried in the blood because the free radical in iron forms a ligand-type chemical bond with O2. Oxygen is dropped off in the cell where it reacts again with free radicals in the electron transport chain complex 4. Here in the mitochondria, oxygen is reduced by all 4 electrons to water by grabbing both the free radicals at once. To do this, the last complex of the electron transport chain grabs the O2 on each end using a free radical for each end. We want to look at how antioxidants work. Antioxidants work in the following manner. The antioxidant donates an electron to a free radical, and the antioxidant in turn becomes a free radical itself. You can see this in several antioxidants. They all follow the pattern of donating an electron to reduce the oxidants. However, in the process, they go from being reducing agents to oxidizing agents themselves. We hear the term scavenger applied to antioxidants, but it is not the way antioxidants like glutathione, vitamin C, vitamin E, and urate work. All these antioxidants are one-time actors. They have a single bullet or electron to give. In some cases, they have 2 electrons. But after they have fired, they must reload. They must get another electron. Dismutases do work like pac-men or scavengers. They don't lose any electrons and they don't need to reload. The way they accomplish this is to use 2 of the reactive oxygen species of molecules and oxidize one to get the electron to reduce the other. Superoxide dismutase uses 2 of the superoxide free radicals. One gets an electron from the other. So the reaction makes 1 O2 and 1 H2O2. Because superoxide is 1 electron from O2, it cannot go all the way to oxygen and water. Superoxide sits 1 electron from O2 and 3 electrons from water. Superoxide dismutase 3 makes O2 and hydrogen peroxide. Catalase is also a dismutase. This means that it does not depend on an electron donor like antioxidants. Also, it acts on hydrogen peroxide. Hydrogen peroxide sits 2 electrons from oxygen and 2 electrons from water. It can safely take 2 electrons from hydrogen peroxide from the molecule that becomes oxygen and give them to the hydrogen peroxide molecule that becomes water. There are three types of superoxide dismutase. Type 1 is in the cytoplasm, 2 is in the mitochondria, and 3 is excreted outside the cell. The importance of catalase can be seen in response of RSV murine models of acute respiratory distress syndrome where intranasal catalase help to shut down the cytokine storm. So that starts to answer the question, why doesn't the production of interferon lead to clearing the virus in cases that develop ARDS? By destroying the AEC2 cells, the virus interrupts the production of surfactant, the production of superoxide dismutase 3 and catalase, and removes the ability for the alveolar epithelial cells to be replaced in a healing process. Having learned how oxygen can be thermodynamically activated and kinetically stable, the 4 redox steps from oxygen to water, how antioxidants work, how a dismutase works, we're ready to get to how ketosis regulates NADPH concentration and why that may prevent ARDS in the next video.